today I'd like to thank you all for coming. First, I'll introduce briefly our experts in the field. We got Vic Thannickal from Tulane and Ana Mora out of Ohio State. They'll be acting as our experts in the field as we discuss Maggie Freeberg's paper discussing the transglutaminase 2 knockout mice. So Maggie did her postdoc. She's doing her postdoc. Initially, she was at University of Rochester, and then she migrated over to VCU, where she is a postdoc right now with Patricia Syme. And then I will let Maggie take it from here.
So big thank you to ATS and the RCF Assembly for inviting me to present today. And thank you, Jason, for the introduction. I also have some exciting news that I'm going to move on to the next stage of my career and start my lab this fall. So I'm really excited to continue this research more independently. And I'm extremely grateful to my mentor, Dr. Syme, who's able to join us today for helping facilitate this next stage. So as Jason alluded to, we're going to talk about these transglutaminase 2 knockout mice and looking at their lung function and bleomycin-induced injury and the metabolic derangements that are associated with fibrosis. So there are a few disclosures related to this work. This work was completed in partnership with UCB Pharma, and 3 of the co-authors on the study were employed by UCB Pharma at the time. Dr. Syme has received consulting fees, and then 3 of my co-authors have intellectual property related to the study. I personally do not have any disclosures related to the study. So I study progressive pulmonary fibrosis, and idiopathic pulmonary fibrosis, or IPF, is a fatal progressive scarring disease with a very poor prognosis. Most patients have 3 to 5 years of survival from time of diagnosis, and there are 2 current FDA-approved therapies that slow progression in some patients, but they're not well tolerated, and slowing progression does not equal a cure. So there's a critical need for improved therapies to be developed, but developing new therapies has been particularly challenging for pulmonary fibrosis due to the complex nature of the disease and the compounding feed-forward loops that are present. So my research really focuses in on understanding the pathogenesis of disease and the fibrotic tissue remodeling and myofibroblast activation, as well as the amplifying feed-forward loops, specifically focusing in on the dysregulated cell metabolism and altered mechanical properties present in disease, and also the interaction between mechanical changes and metabolism. So for today, we're going to focus in on tissue transglutaminase 2, or TG2 as I'll refer to it. It is a member of the protein glutamine gamma-glutamyl transferase family, and the key function of TG2 in this context is that it cross-links proteins, and it specifically cross-links between the lysine residue and the glutamine residue in collagen, and this creates bonds that are highly resistant to proteolysis and degradation, and so it's acting as that reinforcing rebar in the fibrotic matrix, and it also contributes to that stiffening feature with mechanical changes. Now, TG2 isn't only a cross-linking enzyme. It does have other types of reactions that it can catalyze, and this is very location dependent. So in this context, I'm focusing in on TG2 and its role in the extracellular matrix and how it stabilizes the ECM. However, it also has functions at the cell surface, and it can bind to integrins as well as fibronectin. It also has the ability to function intracellularly as a G protein. And in the nucleus, it can act as scaffolding to help with nuclear proteins, histone modifications, and potentially other unknown functions. Now, over a decade ago, Dr. Syme was actually interested in looking at TG2 in IPF patients and had this study that was reported in the Red Journal by Keith Olson at the time, looking at the the presence of TG2 within human biopsies. And what we can appreciate with this immunohistochemistry, where the brown staining indicates positive staining for TG2, is that there are some low levels of TG2 present in normal lung tissue. However, in these 3 different IPF biopsies, we can see a substantial accumulation of TG2 present in the fibrotic lesion of the biopsies. In this study, they went on to look at TG2 deficient mice in the bleomycin-induced fibrosis model. And what we see in the wild type mice with saline controls is the normal lung architecture. And then with the bleomycin injury, there's a substantial increase in cellularity and architectural distortion and fibrosis. And in the TG2 knockout mice, they have very similar baseline levels, but better preservation of lung architecture, and the fibrosis just is not as dense. When they quantified the extent of collagen accumulation with hydroxyproline, there's actually a significant improvement in the TG2 knockout mice with less collagen accumulation with bleomycin-induced injury. Now I'm going to pivot a little bit to the metabolism side of the story and metabolism background. Dr. Syme was also interested in looking at aberrant metabolism and actually identified that lactate is upregulated in patients with IPF, and went on to show that lactate production creates this feed-forward loop where there's extracellular acidification and a decrease in pH that becomes present outside the cells, which is one mechanism by which TGF-beta can be activated as our central profibrotic cytokine. This then feeds back and activates further myofibroblast differentiation and LDHA, lactate dehydrogenase A, which is responsible for that conversion of pyruvate to lactate. And then we see activation of more lactate being exported. Now, the concept of dysregulated metabolism has really grown into this field of fibrometabolism by several groups, looking at the metabolic requirements of pulmonary fibrosis, not just in fibroblasts, but in other cell types present in the lung as well. Here is a very nice picture from a review highlighting that there are glycolytic changes, there's pentose phosphate, serine glycine synthesis, TCA cycle, and glutaminolysis are all pathways that are implicated in fibrosis and feed into the metabolic requirements of nucleotide synthesis and enhanced energy production for proliferation of cells and enhanced migration, as well as collagen synthesis. And so this all feeds into the fibroblasts are more metabolically active to promote the matrix production and be able to proliferate and sustain themselves. So coming into this study, we were really asking the question of whether or not tissue transglutaminase 2 or TG2 and matrix stiffening can interact with the metabolic reprogramming in pulmonary fibrosis. And in this study specifically, we're looking at examining the lung physiology and the mechanical changes that are present in TG2 knockout mice and looking at metabolic alterations associated with that. And I hypothesize that TG2 knockout will have preserved lung function paralleling the reduced fibrosis that we saw in the previous study, and that there will actually be a link between TG2 deletion, reduced fibrosis, and fewer metabolic alterations. And these studies are highlighted in our recent paper in Physiological Reports. Transglutaminase 2 knockout mice are protected from bleomycin-induced fibrosis with preserved lung function and metabolic derangements. So the overall experiment design for this study is purely just a mouse model. We're using the single dose bleomycin model with 2 units per kilogram of bleomycin delivered by oropharyngeal aspiration into either C57 LAC6J mice as our wild type controls, or the TG2 global knockout mice that are our experimental group. We're first looking at fibrotic outcomes, looking at different histological outcomes with trichrome, H&E, and SHG imaging, hydroxyproline to quantify collagen content, pulmonary function testing, and RT-PCR of different matrix-associated genes. Our other outcomes are looking at the metabolic derangements, First looking at immunohistochemistry of some of the LDHA, lactate assays, and then performing untargeted metabolomics on total lung tissue homogenates. So first, just looking at fibrosis development, here is a hematoxylin and eosin stain, very low magnification, just to give you an idea of how the lung architecture is altered in either the C57 wild type mice or in our TG2 knockout mice. We can see in the saline control mice that they have very normal preserved lung architecture. And then with the bleomycin-induced injury, we have a very nice fibrotic lesion and increased cellularity present in this region of the section, as well as in the TG2 knockout mouse. We still have fibrosis present, but there's a reduction in the total fibrotic lesion size. And this kind of parallels what was seen previously. Now here we're looking at second harmonic generation imaging and hydroxyproline to look at more of the collagen-specific changes in the fibrosis model. And looking at higher magnification images with SHG, we can see very nice collagen accumulation as indicated by the green staining here, and destruction of the architecture. Where with the TG2 knockout mice, we see some presence of collagen, but it doesn't get to the same level with bleomycin injury as with the C57 black 6Js. And when we use a computer algorithm in Definiens to quantify the extent of green in those images, we're able to detect a significant increase in the collagen presence in the wild type bleomycin model, but we don't detect a significant change and even a trended decrease in the knockout bleomycin model. So there's less collagen present. And this parallels what we find with the hydroxyproline assay as well. So this is hydroxyproline per right lung lobe, where the entire lobe is homogenized and quantified for hydroxyproline. We detect a significant increase in collagen content in the wild type bleomycin model compared to saline controls, but no significant difference between knockout saline versus its bleomycin counterpart. We next looked at the PCR of matrix-related genes. And so here we're looking at collagen 1, collagen 3, and fibronectin, which are three key fibrotic matrix genes that people typically look at. And similar to what we saw with the collagen, SHG, and hydroxyproline, we have a significant increase in collagen 1 at day 21 of the bleomycin injury, and then a significant decrease in collagen 1, I'm sorry about that, compared to the wild type bleomycin with the knockout bleomycin, and no detectable significant difference between knockout saline and knockout bleomycin. And this pattern holds true for also collagen 3 and fibronectin. So now that we've established that we have less fibrosis, kind of paralleling what was seen in the initial study, we're now expanding the knowledge to look at pulmonary function testing to see if we can get a better granular assessment of the mechanical changes that are present in the bleomycin injury. Now, the first parameter we're looking at with pulmonary function testing is elastance, which mimics the elastic stiffness of the entire lung space. So it's contributions from the lung, the chest walls, and the airways. And now in this model, we are seeing a significant increase in elastance or stiffness associated with the bleomycin injury, and a significant decrease in wild type bleomycin versus the knockout bleomycin. So the TG2 knockout mice lungs are less stiff in the presence of the bleomycin injury. The next two parameters we looked at were dynamic compliance and inspiratory capacity. So compliance is the inverse of elastance, where we're looking at the ease with which the respiratory system can be extended or expanded. And so what we observe in the wild type model is that there's a significant decrease in compliance. So the lungs are stiffer and harder to expand with the wild type bleomycin. Interestingly enough, we detect a significant improvement in the knockout bleomycin, but there is still a significant decrease compared to its saline counterpart in this case. And so we're still able to detect some fibrotic changes with dynamic compliance. And this pattern holds true for inspiratory capacity, where we're measuring the total inspiratory volume of the amount of air that can be held in the lungs. And so we see with the traditional wild-type bleomycin model, there's a reduction in inspiratory capacity, a significant improvement in the TG2 knockout mice, but still a detectable significant change between knockout saline and knockout bleomycin. I was next interested in the viscoelastic related parameters, and tissues and organs, including the lung, demonstrate this viscoelastic behavior where they have time and history dependent mechanical properties that combine both the elastic and viscous behavior of the tissues. And this can be demonstrated with the energy dissipation shift with the loading profile. So when you apply a stress or apply a known amount of deformation and measure the stresses within that tissue, you get one loading curve. And as you unload that tissue, energy is actually going to be dissipated and it follows a different curve. And this can be quantified by the area in between the curves and is termed hysteresis. Now, two of the lung PFT pulmonary function test parameters that give us some insights as to the viscoelastic changes are tissue elastance, which is the energy conservation within the alveoli, and tissue dampening, which is the energy dissipation. And what we see is that there's a significant increase with wild-type bleomycin injury in the tissue elastance and tissue dampening, but a significant improvement with the TG2 knockout bleomycin model. So we're improving the viscoelastic parameters more substantially. So overall, to summarize the fibrosis changes, is we have less fibrosis and improved lung function properties. Now, to get to the meat of where some of the changes are is looking at the metabolic outcomes and how TG2 interacts with metabolism. And so here we're looking at the histology and untargeted metabolomics. So first we looked at LDHA, which is that enzyme responsible for converting pyruvate to lactate, and its presence within the fibrotic lesion of the tissue. So here we have our C57 black 6J wild-type mice. Here's the low mag H&E just to kind of orient you to the tissue, and then higher resolution trichromes where you can start to see some of that collagen accumulation in the fibrotic lesion, and then our LDHA staining. And here you can see that LDHA is present at baseline, but also starts to accumulate in the fibrotic lung. What surprised me was that LDHA is also present in the fibrotic lesion of the TG2 knockout mice. And so we still see very substantial accumulation in LDHA. When we look at the LDHA quantification by total tissue volume, so here we're doing total tissue lung homogenates, of the lobes, we are also able to detect a significant increase in LDHA by Western blot and an improvement or a reduction in LDHA in the TG2 knockout mice compared to bleomycin. Now, I kind of attribute this reduction to the fact that LDHA is still present within the fibrotic lesion, but the lesions are smaller in the TG2 knockout mice, so therefore we just have less total LDHA when we're looking at the entire lobe instead of just focusing in on the lesion. Another portion of the story was looking at total lactate within the lung tissue using a lactate colorimetric assay. And what we see here is a significant increase in lactate within the wild-type bleomycin injured group, but we also detect a significant increase in the TG2. And so there's a little bit of a disconnect between LDHA total expression and then the amount of lactate that we're able to detect within the lung, that the TG2 knockout mice still seem to have this elevated lactate. So I then looked at the total TGF beta presence, because TGF beta was all a part of this LDHA lactate story previously, and found that when looking at total TGF beta, not just active, but total, we see that there are very comparable levels in wild type mice, independent of saline versus bleo, and comparable levels in the TG2 knockout mice, independent of saline or bleo. But when we look at wild type versus TG2 knockout, there is in fact a significant decrease. So the TG2 knockout mice have less total TGF beta in their lungs. Given all of this, we were really interested to look at metabolism beyond just this lactate cycle. And so I next performed untargeted metabolomics on the lung tissue homogenates. And here is just a partial least squares plot to kind of highlight where we're seeing variables or changes within the data set. So in this case, every single dot represents an individual mouse, and they are just statistically clustered based on their similarity of the entirety of the data set. And what jumps out to me is that there is substantial overlap between the wild type and knockout saline animals, and significant overlap between the wild type bleomycin and knockout animals, with maybe a little bit of shift in between the two. But overall, this seems to be driven by injury rather than mouse strain. And when we look at a heat map of what is changing, in this case, each row represents an individual compound that was detected by untargeted metabolomics, and each column is an individual mouse that fits into either the wild type saline, TG2 knockout saline, wild type bleo, or TG2 knockout bleo. And what we see once again is that wild type and knockout mice have very similar metabolic changes, and it's driven by injury. However, we can start to look a little more granularly and see that when we're looking at metabolites that seem to be elevated in injury compared to saline, that there seems to be a reduction in magnitude in the knockout mice. They're just a little lighter in color. So on our scale bar, dark brown indicates high levels and dark teal indicates low levels of that compound. And overall, we see in this column that wild type is darker than the TG2 knockout. It's also important to note that there are some baseline differences in the TG2 knockout mice compared to wild type controls. However, we only had 2 named compounds in this area, and so it's hard to elucidate exactly what the baseline differences are, but there are a few minor changes. They're not exactly identical at baseline prior to injury. So with the untargeted metabolomics, I take the filtered data set of what compounds are changing and named, and then perform pathway analysis or pathway enrichment in MetaboAnalyst. And in MetaboAnalyst, it starts to cluster pathways or the metabolites into pathways so we can start to make sense of the data. And what we have here is a pathway enrichment plot that the magnitude of the bar represents the enrichment ratio, which is how likely is that pathway to be identified given the size of the data set input. And then the statistical significance of that pathway. So the darker the red, the more significant the pathway. And if we take a look at the top 5 pathways that are enriched, it's glutathione metabolism, urea cycle, glutamate metabolism, ammonia recycling, and aspartate metabolism. When I put this together into a pathway, we can see it all kind of converges on glutamate being very central to this process. And we see that glutamate can feed into energy metabolism in the TCA cycle. It can feed into collagen synthesis with arginine and proline metabolism, or it can feed into glutathione and reactive oxygen species. And so next, I want to call out 3 of the metabolites that were detected and changing, but also a little bit different between TG2 and wild type, which are aspartate, glutamine, and glutamate. And what we see here is that in the wild type bleomycin group, we see a significant increase in glutamine and glutamate with injury, and no detectable change between knockout saline and knockout bleomycin. And so this indicates that there seems to be a reduction in that glutamate consumption or production in the TG2 knockout mice. And one hint that we get is that aspartate seems to be increasing in the wild type bleomycin model. And to me, this indicates a potential feeding into the TCA cycle with the energy metabolism, as aspartate can be one of the byproducts of this reaction where glutamate's converted into alpha-ketoglutarate. And so this is a hint that glutamate metabolites in energy metabolism seem to be a contributing factor, or at least an outcome of this bleomycin-induced fibrosis. So key findings from this study are that TG2 global knockout mice are protected from developing pulmonary fibrosis and have preserved lung function. The tissue metabolic derangements in bleomycin fibrosis alter amino acid and energy metabolism, and that knockout and wild type mice actually have very similar metabolic alterations. Most of the metabolism is altered due to injury rather than strain, and knockout mice just are lower in magnitude. And then the untargeted metabolomics really points to glutamate as a possible alternative energy input into the TCA cycle. And that's one of the key features of bleomycin-induced fibrosis. So we're starting to kind of bring together this picture of metabolic changes in TG2 interactions, where I actually think we have parallel circuits that converge on TGF-beta. So if you recall that the TGF-beta or the TG2 knockout mice had a reduction in total TGF-beta. And so I think we're kind of putting a break in this cycle over here, where deleting TG2 reduces the amount of TGF-beta that's present to contribute to the metabolic reprogramming processes that are TGF-beta driven. And so I think that is one way. If we break one aspect of the profibrotic feed-forward loops, it might indirectly interact with the other pathways overall. So I think we just see less fibrosis, but overall the metabolic changes present in fibrosis are the same. So with that, I would really like to thank my co-authors and the Syme Lab here at VCU. Dr. Syme and Dr. Thatcher are incredible mentors, and the rest of our research group is incredible. I want to thank our patient support group. I didn't present any of our human samples today, but the patients are central to our research process, and they actively participate in our research studies with donating blood and exhaled breath and tissue biopsies, my co-authors on the study, and then my funding sources. So with that, I'll take any additional questions.
All right, thank you, Maggie. That was excellent. I actually momentarily panicked on the introduction earlier, and I completely failed to mention the fact that you have multiple first author publications, grants all throughout your career, including PFF and Parker B. Francis, and you transitioned to a K. And I wanted to say congratulations on your faculty position. Next time I'll look at my notes and panic a little less. And so now we can take a combination of questions, as well as I will turn it over to our experts, Vic and Ana, who collectively have expertise in cell signaling associated with disease progression and pulmonary fibrosis, as well as mitochondrial signaling and senescence, and a combined over, I believe it's like 300 manuscripts between the two of them. So we can really see what we can learn between Maggie's paper and these experts, and feel free to ask questions. I believe Sergey is manning the chat box, and we will go from there. If either Vic or Ana would like to lead on their thoughts.
Ana, you want to go first, or?
I can do like a general comment only that I want, of course, to congratulate Maggie and also in general the Symes group, because has been one of the leaders to bring the metabolism arena into this fibrotic and in general lung disease. They really have been pioneers in the field. And I just want also to mention that I think more and more we see how these pathways of metabolism are really relevant, not only for normal cells and communications between different cell compartments, mitochondria, nucleus, cytoplasm, even extracellular compartments like here, but communications between cells and probably, hopefully, a new metabolic like metabolites or enzymes can be targets for novel therapies. And I think that's where Maggie probably will go and probably will be some of the questions. But I just first want to say that this is great work and very good point for what in general, the field is moving to try to learn more about these pathways. Victor, I'll let you and then we can go more in details.
Sure. Yeah, no, I want to also congratulate Dr. Freeberg and Dr. Syme. And as Ana said, they really were the pioneers to describe early on the metabolic reprogramming and fibroblast activation and myofibroblast differentiation. And so, they deserve a lot of credit for that. Now, as we move into the transglutaminase story, which Maggie presented very elegantly, one of the things that caught my attention was the multifunctional nature of this cross-linking protein. So it has extracellular effects, but also functions intracellularly as a co-receptor for integrins and GPCRs. So when you put the story together, Maggie, your focus seemed to be in the extracellular compartment in terms of the stiffness-mediated effects. And I think you were explaining and interpreting your data primarily downstream from that activation of TGF-beta and all of the other things, the profibrotic activities of TGF-beta. I was wondering if there's a possibility that some of what you're seeing may be due to the sort of non-canonical functions of TG2 and functioning in glutaminolysis or other things. And, we can come back to the very interesting phenomena that we see in biology, where there is this balance between energy requirements, energy balance, and biosynthesis. And, with fibrosis being as complicated as it is, there's obviously biosynthetic pathways related to extracellular matrix formation and collagen production, etc. But also these are highly energy dependent processes. So you're balancing, the cell has to balance this, energy production and the biosynthetic pathways, which are competing pathways. Maggie, your thoughts on, some of the, is it all related to the matrix, or are other ways to explain this? Since you knocked it out, you basically cannot dissect between the intracellular functions and the extracellular effects.
Yeah, so that is a fantastic question, especially given the multifunction nature of TG2. We did not directly look at it in this study. We do have another study where we actually, unfortunately, so part of the UCB collaboration is they're developing therapies against TG2. And so we looked at it in other preclinical animal models of rat fibrosis and saw that when we inhibit the extracellular activity of TG2, and I misspoke, not rabbit, but when we inhibit the extracellular activity of it, we actually are able to see a reduction in fibrosis. So coupling this paper with that data indicates to me that we are at least looking at some of the fibrotic related outcomes being reduced due to that extracellular activity. In terms of the metabolism, I don't know. And I would speculate that some of those baseline changes that we're seeing are actually related to some of the other functions of TG2. But I don't entirely know how they are connected. One thing I do want to highlight is TG2 activation is somewhat calcium dependent, and calcium signaling in myofibroblast differentiation and activation is also intertwined. And so I think you raise a very good point that I think it is absolutely the multifactorial processes. Trisha, do you have some thoughts?
No, I agree with everything you're saying, Maggie. But we have some great question, Victor. We have some mutants that are devoid of catalytic activity, that are devoid of nuclear translocation signals, to begin to tease out that very question. So we've done some work in vitro, much more difficult to do in vivo, but it's a great question. There's a lot of literature in oncology and the role of TG2 and increasing kind of recognitions. There's nuclear functions of TG2. I think we don't really understand at all how that might be related to fibrosis, but it's a bit this commonality of lots of pathways, and oncogenesis are common and recapitulated in fibrotic tissue. So I think there is a lot of unanswered but important questions there, and we have a few tools to help us start to parse out some of those things, at least in vitro.
So probably another question is because you have a whole body knockout, right? If you see effects not only in fibroblasts by this enzyme, because we know TG2 is also involved in inflammatory responses or proliferation, for example, etc. Or even if you know that, I know in previous prior publication, you know that definitely if these fibroblasts that lack TG2 are less active, etc. But if you have also examined maybe some of the other responses not related 100% to fibrosis.
Yeah, exactly. So the global knockout is a tool to kind of look at the development of fibrosis, but yeah, we're definitely affecting other cell types as well, and having that interaction is compounding in this model.
So,
And yeah, go ahead.
no, I'm just gonna. Let me just ask you in terms of metabolism, linking the story to the metabolic analytes that you've discovered and the upregulation of those specific metabolites that you mentioned. Do you think that is downstream? So the aspartate, glutamine, and glutamate. Now, one of the, as you know, the limitations of these types of measurements, which are beautifully done, and I know that VCU has one of the really outstanding cores in measuring these metabolites. One of the limitations, though, is that it gives you a picture at a static point in time, right? It doesn't tell you the flux of these metabolites through different pathways. It doesn't tell you how quickly the TCA cycle or this turbine is churning, right? It doesn't give you the rate of oxidative phosphorylation. It may be that these metabolites are accumulating because they're not being used, which would be the opposite of a revved up system where you're producing more of the upstream metabolites. So which way is, how could you determine which of those is true in your system?
Yeah, so you're getting to some of my ongoing research. Is we are doing some active seahorse flux assays, not just with like TG2 deficient cells, but looking at just like primary fibroblasts and modulating other aspects of the fibrotic activators. But so yeah, we're doing seahorse assays to look at substrate dependence. So you can look at the glycolysis or glutamate dependence of the flux of the assays. I'm also doing some glutamine tracing stuff where we can put in stable isotope labeled glutamine into the system. And in cell culture, it's fairly simple, where you can remove the non-labeled glutamine and trace where it goes and how quickly that happens, and kind of look at the flux of the system. And so this is an area of active research, is trying to understand how glutamine metabolism is contributing to fibrosis, not just in the TG2 context, but in response to changes in stiffness, in mechanosensor activation, and then just general pro-fibrotic cytokine stimulation as well. And so understanding the different metabolic changes that occur and how glutamate is contributing. Does it all just feed into collagen synthesis? Right now, I'm starting to see hints that some of it also feeds into the TCA cycle and energy metabolism.
Right. And so is your experimental variable in those planned future studies, is that the TG2 that you're going to manipulate levels of or activities of, or is it the TGF-beta?
So for the most part, I am actually separating a little bit from this and looking into just stiffness mediated changes. And so we're looking at how the mechanical environment alters metabolism. So we use the TG2 knockout model in this case to look at changes in stiffness or kind of adjacently assess that in this context. But in our cell culture studies, I'm doing more just mechanical activation and then comparing to TGF-beta. And then we can also knock out or knock in other components that we think are pro-fibrotic.
Yeah. And the reason I asked that question is, again, is this, as you showed in your summary figure, downstream of TG2 affecting TGF-beta signaling, or is it an independent effect of TG2 on glutamate metabolism?
I don't entirely know. I don't know. It'll be interesting to see, and maybe we should consider isolating some cells from our TG2 knockout colony and just looking to see how the metabolism and flux changes with that. Right now, I've been focusing my flux experiments on primary human fibroblast cells, but we could definitely leverage the mouse tools that we have as well.
So maybe another question using particularly the human fibroblasts. We know that has been described as senescence, right? In these fibroblasts, at least in some of these types. Have you analyzed the association between glutamine and the senescence phenotypes, or these also different now subtypes of human fibroblasts that has been described and are probably, I don't know, CTHRC1 is one of these fibroblasts is related to this cross-linking of the function of the enzyme? Or?
Yeah, so that's a really great question. I think we have not directly looked at it. And I don't know how, like thinking about the metabolic pathways and what I know about senescence, how they would directly interact. But I imagine that we've seen this glutamate metabolism pathway be hits pretty regularly in our mouse models, our cell culture models, and particularly in our profibrotic fibroblasts. And so I don't know if it's chicken or the egg, but I imagine it goes concurrently with the senescent apoptotic resistant fibroblasts that accumulate in fibrosis.
Have you seen changes in the expression of TG2 with age?
That's a good question. I have not looked. Trisha, do you know about that?
No, great question. I've not looked. Haven't seen anything published.
Mm-hmm.
May have missed something, of course, but I haven't seen anything.
Yeah, we should get great questions and great discussion. We should get some people who are on the call. If you have any questions, please put that in the chat or unmute and raise your hand. It looks like there are mostly senior people on the call, but let me just say, if there are any graduate students or other students in the audience, one of the beautiful aspects of this biology and biochemistry is exactly as Maggie said, is that cell states and cell fates, several of which we've alluded to, is really linked to metabolism and energy, how it's utilized. I mean, there's basically two pathways of everything. It boils down to catabolic and anabolic processes, but the cells, due to the signals, intracellular and extracellular signals, intercellular signaling, all of this sort of converges on a decision that has to be made by cells. Either they're going to turn on biosynthesis, or they need the substrates, whether it's glucose, hopefully they don't have to rely on lipid metabolism as much, or protein catabolism. But the point is that these cells have to decide whether to proliferate or differentiate, and those come with a specific set of requirements for macromolecular synthesis and function. So this is known as anapleresis, and the Greek word, I don't know if I'm pronouncing, ana for up, pleresis for fill. So you're filling up the TCA cycle with these, carbon-based molecules that then have to be catabolized through the TCA. But in situations where you can divert that to collagen, you then, may be limiting the cell's ability to make ATP through the TCA cycle. So anyway, anapleresis this is one of the classic pathways for anapleresis. And a lot of it is dependent on extracellular signals that are present, whether it's the stiffness, mechanical signals, or soluble receptor-mediated signaling through TGF-beta. Sometimes it's hard to know, and it's all happening at the same time in a fibrotic lung.
All right, looks like we have a question from the audience. We got Scott McCall here. He's a faculty member here at Vanderbilt. Scott?
Maggie, awesome talk. Thanks for sharing that. I guess my question was actually going to be a little bit more nuanced, just in terms of kind of parsing the cell compartments a little bit, just because obviously, fibroblasts behave a little bit differently even than epithelial cells when they proliferate, and even, a lot of the immunometabolomics field sort of suggests a lot of this glutamate is really very cell type dependent. And I guess part of what I was curious about is from kind of the extant data that you have and looking through this in the bleomycin model, as far as the way in which you feel like this pathway might be modulating some of the different cell compartments.
Yeah.
Just because obviously that's a big component of the way that you could interpret this bulk work.
Yeah, that's a fantastic question, and I don't have a great answer. Obviously, if we're looking at a total lung tissue homogenate, we're seeing a general shift, but I also think some of the less statistically significant trends that we see is because we definitely have compartment specific changes that are diluted out by digesting the whole lung. To kind of answer this, I'm working on some spatial metabolomics. Unfortunately, spatial metabolomics isn't quite at the resolution of individual cells yet, but we can at least get pathologic annotations and about a 25 micron size field. So hopefully I will have a better answer for you in about 6 months as to which compartments are really driving these processes. But that's how I'm planning to elucidate some of that, is looking at the spatial metabolomic aspects of IPF and mouse models of fibrosis.
So based on that question from Dr. McCall, do you think that the TG2 affects in epithelial cells? Does anybody, Abraham or anybody else on the call, want to predict what might be happening in epithelial cells or immune cells, as Ana brought up?
So I imagine in immune cells, I can better predict there, I think, than the TG2 role in epithelial cells. But in immune cells, I imagine that it is affecting some of the cellular infiltration and migration, which we think is definitely a key component of the bleomycin-induced model specifically. And so if we looked in a different mouse model of fibrosis, we might see a slightly different pattern. And so that's why I'm emphasizing, these are bleomycin changes. And so I imagine it's affecting the infiltration of those circulating macrophages and whatnot. Epithelial wise, I don't know. Does anyone have any thoughts?
Not really, but in this context, of course, but I say it's not that in cancer, right? It's increased and so promotes probably in general glutamine is affecting proliferation, is affecting autophagy because there are levels of amino acids. It's glutamine, of course. I'm talking about, we don't know transglutaminase, how it will be completely affected. And that's a question I have for you. How are you planning maybe? I know there are inhibitors for the extracellular versus the intracellular roles of transglutaminase. Are you planning to use some of those maybe mechanisms or some genetic compartments, and also if it's another enzyme maybe that goes through the metabolism of the glutamine, also important to know
So I'll
how
say
it metabolism.
lies in more looking at elucidating the metabolic changes associated with mechanotransduction and the mechanical changes. So I'm going to shift away a little bit from the TG2 focus on it. But if I was to continue the TG2 focus, it would definitely be moving towards trying to find different inhibitors for those different compartments to separate out the function, like what you're talking about. And the hard part is TG2 doesn't get activated as easily by fibroblast in cell culture, and so you can't just do standard tissue culture plastic cultures plus or minus TGF beta to study TG2 related functions. It's a little bit more nuanced and complex for doing that, but looking at the different compartments and being able to block either translocation like Dr. Syme alluded to would be a way to kind of separate out those mechanisms.
Or even work with the receptors or transporters, right? That goes one that goes inside the cell, another that goes into the mitochondria. Although I think that is not well known, some of them, but would be great ways.
Yeah.
So Maggie, since you brought it up a couple of times, let me ask you a question about the matrix stiffness. So let's take TG2 and TGF beta out of the equation. Mm-hmm. Just look at soft and stiff matrices. Yes. And look at how fibroblasts respond to them. Which of these metabolic pathways are upregulated and how? Do you know, or is that?
So that is also a paper I am working on currently. When I look at the differences, there are dramatic metabolic shifts in fibroblasts when you seed on physiologically healthy or fibrotic stiffnesses, and then even more dramatic when you compare to standard tissue culture plastic conditions. I am seeing a lot of pathway overlap. Normally there's a little bit of nuanced changes, but most of it's central to that central carbon metabolism with glycolysis, TCA cycle, and glutamate metabolism in these fibroblasts. I also see aspects of arachidonic acid and some lipid metabolism changes. But I think we're seeing very much so a convergence of, if I jump back to the fibrometabolism pathway, we see less so of like the pentose phosphate and hexose biosynthesis and more things related to energy and collagen production. And if we think about what happens when we seed fibroblasts on matrices that are changing in stiffness, is you're going to change their proliferation rates. We all know that if you try to expand fibroblasts on soft substrates, or you may not, they don't really proliferate as much. And so we're capturing some of those changes. They also do not spit out as much matrix. And so I think those are some of the central pathways that we're seeing related to that.
So isn't differentiation and proliferation sort of antagonistic? Mm-hmm. And if that's the case, you're saying that stiffening is required for proliferation, but you're also saying that stiffening causes them to differentiate more. So how do you reconcile that?
I reconcile it by the fact that I am measuring bulk analyses on wells and I am not looking at individual cells.
Oh, wow. So cells are doing different things under the same conditions. Okay.
And so I don't know if they're kind of in flux. I agree that they're not doing both things at once, but we might see some proliferation processes. A lot of times I am looking at cells after 72 hours in condition to look at the effects of being on substrates for that long. And so we're somewhat capturing endpoint differentiation metabolic changes rather than immediate ones. I'm also looking at trying to manipulate different metabolic enzymes and try to capture some earlier time points to see how that might be contributing. But yeah, I think ultimately we've got a heterogeneous population of fibroblasts. I start with primary cultures that we isolate from lung tissue biopsies. And so even the fibroblast, we don't have clonal populations. And so we have different population subsets within our conditions. And then they might do slightly different things in response to the mechanical cues. Dr. McCall, you had another question?
Oh, I think you're right, and in part, I also wanted to just I think kind of chime in on Dr. Thannickal's point here. I'm not sure that proliferation and differentiation need to be thought of as mutually exclusive here. In part, just because a lot of these pathways, from a biosynthetic perspective, you have to both be able to make purine nucleotides and pyrimidine nucleotides and all those sorts of things as part of a lot of the sort of underlying metabolic pathways required to function and differentiate and cycle lipids and do all the sorts of things that we think of as a differentiated cell, in addition to actually being able to proliferate. And so I will say, in part, this is driven by some of the things that we're finding in our systems also. But I think we've sort of tried to work at this proliferation, which I hate making words like that, but at the same time, I think on some level it's not a wrong way to think about it. And I think even Dr. Mora's recent paper sort of suggests that these are all also sort of part and parcel to one another, that disrupting underlying cycling and mitochondrial function and metabolism really goes with both proliferation and the ability to functionally differentiate. And so I wonder if down the road, that's really how we'll sort of think about these processes in terms of building on each other, rather than you either can proliferate or differentiate, but rather being good at both probably is requisite. And that was just all I wanted to contribute, because I think Maggie, your work sort of really speaks to that.
Thank you.
I just want to mention something is to really analyze this. You will be needed bring kind of epithelial cells. We have these progenitor cells, and they go to differentiation, transitional cells, etc. That you will need maybe at one point, I don't know, work with your fibroblasts to try to follow up. But independent of that, so we have a proliferative, of course, the progenitor cells needs to proliferate to maintain that. But differentiation, I agree with Victor. At one point, the chromatin, right, needs to remodel, and there are certain stress that make the cell pause. And we see senescent, at least phenotype, in these transitional states. But is something that we see, but it's transitional. It's not. Can we call that full senescence, or it's just a pause that needs to be before going to full differentiation? But I think more studies would be needed to continue with this discussion. That is really exciting, and I really thanks Dr. McCall to bring it, and Victor, because it's something that we are all working on, trying to understand how these two processes go in this process of during repair, etc., normal repair of this repair, etc.
I thank you all for your insights, and you bring up fantastic points and experiment designs. And I wish I could just run untargeted metabolomics on everything all at once at all these different time points and single cell populations. I just don't think, like, some of it is technology limitations. Like, if we had the same resolution to be able to do metabolic profiling on cells the way we do with like single cell transcriptomics, that would really transform some of our insights into I think we're all in the right buckets, that the metabolism is going to change rather quickly to fit these demands. And when I'm measuring things that are over days, I'm going to miss some of those key pathways. And so how do we hone in, especially if you're harvesting for metabolic analyses? Everyone says metabolism changes so fast. Like, we can't even flow sort the cells because the metabolism changes so quickly. Whenever I'm doing harvests for from mouse tissue. We have to do it really fast in comparison to other processes. And so if it changes so quickly, how do we capture it in our studies? And so I think there are some technological limitations that we kind of have to overcome over the next few years to really get to those questions.
All right, I think we are out of time. I wanted to jump in and thank Victor and Ana for the conversation and discussion. Maggie, great work as always, and we look forward to seeing more great work in the future as you transition to your new position. Thank you all for coming.
Thank you, and see you all at ATS.
Yes.
Yeah.
Yes.
Bye-bye.
Bye.